So I'm going to hand over now for the panel discussion.
So it's my pleasure to introduce our moderator today, Melissa Miller, who very kindly on short notice agreed to step up and moderate the panel on behalf of our original moderator, Manolis Dermitzakis, who is unable to be with us in person, but as you can see, fortunately is able to join us remotely and will be part of the panel. So Melissa is the Director of Human Genetics in the Internal Medicine Research Unit at Pfizer, where she and her team are responsible for applying human genetics and omics data in the identification and validation of new targets for cardiovascular and metabolic diseases. Melissa is a member of the ICDA Pharma Council and is involved in several other large pre-competitive efforts, including IMI Litmus, AMP-CMD, and the UK Biobank's Exome Sequencing Consortium and Pharma Proteomics projects. So without further ado, Melissa, I'll hand over to you.
All right, thanks. And I'm really grateful that I did not have to pronounce Manolis' last name, which I have been nervous about since last night. You've already met Tuuli and Kristin. I'm going to let Kristin introduce our other two panelists, and then I will start the Q&A session.
Okay, thank you. I would like to introduce two panelists here. Shyam Prabhakar, I've already introduced him as a leader of the Genetic Biodiversity Network in the Human Cell Atlas, which I'm a member of that team, as is Sophia. He has a rich history in being part of the Human Cell Atlas Asia Network and the Asia Immune Diversity Atlas, and has a lot of experience already setting up sites. They've been largely blood sampling to date, but he can speak to tissue sampling in addition to those sites. They've done a lot of setup of the type of networks that Tuuli was talking about, localized hubs for data processing, sample sharing, a lot of consideration that he can address about data quality and reduce some batch effects across these sites, and they have some beautiful data already produced from those projects. Sophia is at the University of Miami. As I mentioned in my talk, she has one of the CZI Ancestry Network projects, so with a strong tissue focus. She has sampling sites in Miami, which is with a very diverse cohort, sampling sites in the Caribbean as well, and several sampling sites in Africa. She's currently in the process of doing the same, of setting up molecular labs in these sites so that sampling, so that data generation can be done locally, and sharing and training sites. And she's very successfully got this project up and running within a year, and it's collecting data already. So she's looking at multiple tissue types as well, with reference normals as well as a disease focus. So please, feel free to ask them many questions. They are the people and the sites that we hope to leverage, along with our colleagues here from the Latin American Genome Project as well.
Okay, so the secret to moderating one of these panels is to moderate with somebody like Kristin sitting next to you, who just gave me a number of easy questions. So actually, Shyam and Sophia, I'm going to ask each of you to talk a little bit in turn about some of what Kristin just talked about. So Shyam, I'll start with you. Can you tell us a bit about the work that you are doing, the cohort that you've set up, how you're doing the sampling, the batch effects? And I'm hoping when we get done going through this, Ricardo will have answered some of your questions as well.
Great. Maybe I'll jump straight to your question first, which is we are doing multi-ancestry sampling in Human Cell Atlas Asia. So one of the projects I'm representing here is Asian Immune Diversity Atlas, which is blood, admittedly now, PBMCs, but multi-ancestry in Asia. And actually, even within individual Asian countries, you have diverse ancestries, right? It's not like all Asian countries are completely homogeneous or even partially homogeneous. So I hope that addresses one aspect of your question. And then going back to the broader question of what's been happening in Human Cell Atlas Asia. So we got started 5 years ago, I think, when Piero Carnenci and Jay Shin launched HCA Asia at a meeting with precisely the goals that Kristin and Tuuli and everyone else has been talking about here, which is how do we diversify, in that case it was a human cell atlas, but more broadly, how do we diversify genomics, right? And and initially it was just, okay, we need to diversify, but we had no project. So then finally we settled on IDA, Asian Immune Diversity Atlas, as our flagship initial project to turn those ideas into something concrete. And that means to have some concrete output. So what IDA is doing is doing single cell analysis of cohorts across Asia, focusing on PBMCs because it's the first place to start, where you genotype each individual and you do single cell RNA-seq on each individual from the blood. And you do it initially, phase 1 of IDA is 5 ancestries across 3 countries. And phase 2 will be at least 8 countries and about 19 population groups or ancestries, whatever you want to call it. So I obviously face the same challenges as, say, GTEx or any other consortium, simply setting up all the agreements, disbursing the funding to everyone, homogenizing protocols across all the sites, training, and simply the logistics of communicating and coordinating across so many sites, and the contracting and so on. So those, I think, will be familiar to anyone who's done cohort studies, and we were no different, except that this was very, very multinational. And so then in each country, you face slightly different, each country has different scientific motivations, a different local funding environment, different objectives, different ethics regulations, different ethics norms, and so on. So how do you find the common denominator and still deliver something coherent and useful is something that each consortium will have to address on its own, but I think we learned a lot there along the way. Batch effects. Batch effects, I would say what we learned is it's not a yes or no. It's not like, do batch effects exist or do batch effects not exist, right? The reality is batch effects will always exist, regardless of what you do. The question is, can you minimize them? So it's continuous. The more you minimize batch effects, the more you can learn from the data you collect across the world. And so we definitely said we'll use exactly the same reagent kits, the same machines, the same detailed protocol, and in some cases, purchasing reagents from the same lot number from the same vendor and tell them, please book this lot number for us. We're going to use it across the countries in IDA. We still had batch effects, for sure, across countries. But they're relatively small, so there's a lot we can learn. For example, we can learn male-female differences, because there you have an internal control within each batch, within each country. You can learn age effects. Intra-country, if you have ancestral diversity, you can learn that. And then you can do discovery validation kind of analysis, where you discover something in one country, and then you validate it in another country. And there the batch effect bar is a little lower, right? If your batch effects are moderate and mild, you can still discover in one country and validate in a different country. And then, of course, the EQTL analysis, you can take a country as a confounding factor, a lab as a confounding factor in EQTL analysis. So the message for us, batch effects for us, was that minimize them as much as you can with all your strength, and then do your data analysis in a way that accepts that they still exist, but you'll still learn something. And yeah, I'd be happy to talk about what we learned also, but it's taken up a lot of time. So.
All right, Sophia, let me ask you the same question and tell us a little bit about your cohort as well.
Right, good morning, everybody. So, Ricardo, I also want to say that for our studies, in the context of diversity, in Miami, for example, 60% Miami City or county, 60% of our population is Latino, Hispanic. The other percent is European and Black, from which 1/3 is Afro-Caribbean. So we have a really diverse population in Miami. And then for our study that's funded through the Ancestry Network for CZI, we're doing a single-cell analysis on fallopian tubes, on breast and prostate, of course capturing blood also. We have a large metadata survey that we had previously adopted and developed across a prostate cancer consortium and African Caribbean cancer consortium, from which I'm one of the leaders in that group. And so in the Caribbean, we're in Jamaica, Bahamas, and Haiti. And in Africa, we're in Kenya and Nigeria. And so we're 6 sites, and the goal is to understand normal across the African, these groups, African ancestry, because a lot of the work that we've studied is looking at cancer, and of course we know people of African ancestry, or I would say people who are self-identified as Black in the context of the US and in the West, have developed disease early, aggressive, and so what about the normal biology is leading to that in different populations.
Thank you. I'm just going to look around and see if we've got any questions in the audience. Cecilia.
This one coming.
Oh, coming your way.
So, really exciting to hear about your efforts, and it's amazing to see the diversification and the knowledge and expertise and exciting projects you already have ongoing. Just taking a step back and thinking about Ricardo's question a little bit, and because we have Manolis live in from San Francisco. I wonder if you, Manolis, because you have worked with Tuuli and Kristin for a long time, can just talk about your experience of it's a very exciting and sexy question, your experience around doing the multi-tissue collection out of a large study with thousands of samples, and how feasible is that to do in any kind of setting, and what setting do you need in order to make that realistic and viable as a project? Because I think maybe that would set the stage for some of the questions asked.
Yeah, thanks, Cecilia. I'll try to answer the question. I'm not sure I can answer the full question. Again, apologies for not being there, but the realities of today are a bit too complicated. I think that remembering from GTEx, and I was actually involved in the workshops of GTEx set up by NIH back in 2007 and 2006 and so on. It's a very complex process to put something together that will be acceptable, will be relevant, and will be considered scientifically valid in such a heterogeneous environment. I think Shyam mentioned all the different sort of challenges, so the technical challenges of the heterogeneity. I think that it's probably much more difficult to think of the aspects of tissue collection and the ethics and the consent. That's a more difficult exercise than the scientific exercise of dealing with heterogeneity. And I do remember long debates about how exactly the consent is being done, what's the success rate, how it's heterogeneous even within the US, and how it's heterogeneous depending on the team that was actually responsible for putting together consent. So I think that finding, again, using Shyam's word, finding the common denominator on the community side, it's going to be a challenge if we want to make this a global collection. But I think it's a very desirable goal. Heterogeneity, of course, has genetic effects that we all care about. There is, but as Tuuli mentioned, there's a lot of environmental diversity that we do care about. And I think we have to admit that, and I'm coming from a personal bias where EQTLs has been 15 years of my life. I think we need to move past EQTLs, and we need to pass just thinking about genetics as the primary reason why we're collecting all this information at the population level. There's a lot more things we can learn about biology. There's a lot more modulators at the population level that if we learn the environment and the impact on the organism will actually help. So I would say that it's a highly desirable exercise that we should engage as soon as possible, but we should recognize that cannot be as quick as collection from Europe and the US, and the one should not delay the other.
Hi, I've got a quite technical question, either for Kristin or Tuuli. I'm sure you've had discussions around this, but what's your thinking around like the number of cells per tissue, how deeply you want to sample, the number of different sides from the tissue, as you were talking about, the whole cartography of a tissue, and the balance between the number of individuals. Do you go for more individuals, less tissue sites, or do you go more broadly across the tissue, more, sites, let's say, in the intestine, but less individuals? How do you see striking a balance in this aspect?
I guess the short answer is that it depends. I think that since BodyMap is driven by geneticists and kind of like still kind of being able to map genetic associations is something that is at the core of BodyMap. I think that we need to make sure that we have at least this kind of like couple of hundred samples to be able to do genetic analysis. So that is, I think, somewhat of like there's a little bit of a lower bar in terms of how low can we go in terms of sample numbers that are still meaningful. And in terms of numbers of cells per tissue, I think it will depend a lot on the tissue. And a little bit sort of like, for example, if there are rare cell populations that are really important, for example, for a disease of interest, then you need to sample more deeply to be able to actually get a representation there. But also having multiple donors for any given tissue kind of helps, because you can kind of like pool the data and leverage that as well. So
I think I'll add that it's probably a little bit determined by the study and what we're doing as well. So let's say, for example, if we're working with Sophia's sites where she's collecting breast tissue and prostate tissue, we won't have the luxury of going anywhere we want and multiply sampling, because these are living donors. So it'll be wherever the site is that she's able to collect that. So it will be a limited site, but it will be the potential for a larger number of donors. We'll have a donor addition there. Whereas for sampling primary tissues, there's simply no way you can effectively sample an entire organ and multiple organs all at the same time and preserve the tissue. However, as Tuuli mentioned in hers, we're going to leverage the existing efforts. So the Human Cell Atlas is very far along at this point, and we have some ideas for organs like the lung and the heart where there are redundant tissues, where we might want to sample to maximize diversity or maximize functional effects of tissues and that we care about. So I think we can learn and leverage from that. Where when we have the choice of where to sample, we can leverage that information to decide where are the best places to sample.
This is Jonathan Pollack from NIDA, and the question I have is, what are the technical obstacles for using tissues on histological slides, where there's probably hundreds of thousands of samples that one might be able to obtain in terms of getting the type of data and the type of omics data that you would need for this project?
I guess there's a couple of obstacles. They're slightly being overcome, which is there are increasing numbers of single-cell technologies that are working with banked old histological sections. They're imperfect. Some of them require, instead of just sampling all of the available material that's there, a lot of the RNA is degraded, so you have to use selection processes to select what you're after. So they're moving in that direction technology-wise so that we can open access to some of these. The other thing is that the vast majority of tissue blocks sitting in banks right now are from pathology, and so they're from disease samples. GTEx was unique in the fact that we created pathology blocks for normal tissues, so that's pretty rare to do. So there is a bit of a challenge, and there's also where for most of those you're missing the environment and the other depth of data that we're really interested in adding to this, and we're missing the genotypes. So, do you have anything to add?
No.
So I think this is exciting. You're proposing a rather complicated design, as I'm trying to get, getting my head around it, thinking about sort of the core that you have on the people with the postmortem, and then bringing in these tissue sites, which you've talked about, you're going to have very different locations and all these different people. And then I think Manolis brought up a good point of how do we move past the EQTL study of what you can get out of all of this information. And this is just a naive question on my part. Like, where are we methodologically in order to be able to really think about analyzing this type of data? And beyond the sort of batch effects, what are you seeing as sort of the key things we need to be able to do that we can't do now methodologically to be able to really capitalize on the type of data you're thinking about collecting?
Well, maybe just to mention one or two things. So obviously the sort of like multi-omic integration and batch correction, and those methods are developing very, very fast right now. And I think that by the time BodyMap would actually have data, that we would be much further along there. What I'm personally very excited about is the ability to use molecular profiles to derive kind of endophenotypes of the cells or the tissues, and then use that as a phenotype in GWAS. For example, we have a couple of papers in bioRxiv from my lab doing this to kind of quantify or infer transcription factor activity actually from GTEx data, and then do GWAS for that, and also kind of this sort of like multi-omic sort of kind of axis of variation that one can then relate to genetics and also environment, other sort of physiological data that we have from donors. This is in the context of TopMed and MESA. So so I think that's one thing that I think will be quite valuable.
I think before we take another question, I think Shyam, you also wanted to weigh in on the histology question as well.
Yeah, just quickly, I think with Kristin or Tuuli made an important point. You also need a lot of metadata and genotypes to do this kind of analysis, which some of the bank samples don't have, but some of the bank samples do have that information, so they can be very valuable. Then it's a question of, does the assay work, and how well does it work? So you can do single-cell analysis now on FFPE samples, but the data quality is not the same as single-cell on fresh samples, especially because the fixation damages the RNA, right? So that's a major limitation, and you'll have a profound technical variability when you're doing single-cell analysis or single-nucleus analysis on these FFPE samples. What you can do then is spatial omics. So spatial omics technologies are really revolutionizing the information we can get out of FFPE samples. But the FFPE spatial omics technologies now primarily they're gene panel based. They're not unbiased. They can't tell you the whole transcriptome. So there's a lot of value, omics value we can get from FFPE samples, but limitations as well. You know that they can't tell us everything we need.
I actually have a follow-up question. I think this could go to almost anybody. A number of you have mentioned the importance of collecting environment and going beyond genetics and thinking about environment. In terms of, are there specific environmental factors that you think that we should be collecting as part of BodyMap? Yeah, absolutely.
Yes, so in our extensive survey, a lot of the questions that we're asking, or part of some of the questions we're asking is, where did you grow up? So within the country, did you grow up on a farm, near a farm? Where do you live? Clearly, how long have you lived where you live? That's very important, especially we know that people move around and they take their genetics with them, clearly, and their risks. But if where you're staying increases that in terms of environmental exposure. We have a really in-depth, of course, a recall, but diet, what are you currently eating? And then family history.
Can I suggest Shyam address that as well? Because in the context of the Genetic Diversity Bionetwork, we've been thinking about it across everyone who's participating in that, and it's a challenging question because it's different depending on where people are.
Exactly. I think the environmental variables, the important environmental variables depend on the environment. So kind of paradoxically. So it really depends on what question you're asking in each place. We have tried to zoom in on a shared set of environmental variables that we should collect across the globe in all places. And it's just difficult. Some consortia will say, oh, we simply do not have access to that information. Or some other places will say, okay, if you look at metadata like medical history or something, it's unethical to ask certain kinds of questions of your donors. So I think the basics we go for at least are, at least within IDA, we look at your exercise, your smoking history, this is not environment, but your drug regimen, your prescription drugs, medication, and exercise, and drinking, and sleep. Those are the things I remember off the top of my head. But I think it's just an open scientific question, which are the most influential environmental variables? If you just look at molecules as a readout or a cell state as a readout, which environmental variables affect your molecules and affect your cell state the most? I don't think we know the answer. And so we really have to explore a whole range of as many environmental variables as we can collect them, do the science, and then eventually when efforts like the ICDA become more mature, we'll know the answer of what environmental variables we really need to collect, essentially. One key insight we got from IDA is that it's not just genetics, and I think Cecilia was also saying this, someone else was saying, because the genetic relatedness does not fully explain the gene expression or cell state relatedness we're seeing in our data. Sometimes the cell state differences are very different in two groups that are genetically most similar. And then you say it's culture, environment, lifestyle, something like that.
May I say something on
I
this?
think, in the interest of time, we'll need to wrap up now, but I hope the conversation will continue over coffee and at every opportunity. So I just want to say thanks to our panelists and our moderator for a great discussion about the vision for taking the BodyMap project forward, as well as some of the technical and logistical issues that have to be taken into consideration. So thanks for sharing your insights from the working group and how you hope to take the program forward. We do have a coffee break now, and we'll be back at 10:30, half past the hour, in order to hear from the disease map team, which will also be great. So thank you to everyone, including our online participants, for your engagement in the BodyMap session. Thank you.
Thanks again, everyone. Thank you.